The overall goal of this experimental protocol is to evaluate how quickly and accurately an individual can respond to specific types of vibrotactile stimuli.
This method could help answer key questions in the rehabilitation engineering field, such as what type of sensor information can be communicated through vibrotactile feedback, and what is the best way to represent sensory information through vibrotactile signals. The main advantage of this technique is that it is very flexible for different biofeedback applications and can be easily adapted for use with a variety of biosensors, such as pressure sensors for measuring reaction force or goniometers for measuring joint angles.
Though this method provides insight into how people respond to vibrations applied to specific areas of the thigh, it can also be extended to other areas of the body. By testing different tactor configurations, we can better understand what types of information can be communicated through biofeedback.
Demonstrating this procedure will be Sam Shi, a graduate student in our lab.
Begin the motor calibration by connecting the large microcontroller board to the computer using a USB port. Within the original microcontroller software, click the Upload icon, denoted by the circled right arrow, to upload the custom script motortest.ino to the board using the USB connection. Then, connect the Z-axis output of the tri-axis accelerometer to one of the analog input ports of the smaller microcontroller board. Connect the positive and ground leads from the accelerometer to the 5-volt and ground ports of the microcontroller board, respectively. Next, mount the accelerometer to the vibrating motor, ensuring that its Z-axis is orthogonal to the flat surface of the motor, and then place the motor on a hard surface.